let me first start by expressing my gratitude to the organizers, particularly Professor Minich and also Professor Stoka for inviting me to present some of our studies. My group at the National Institute of Chemistry works, basically we are interested in molecular mechanisms of NLRP3 inflammasome, but I will not bore you with molecular and cellular details, so we'll present a rather simple study where we designed peptides to stop NLRP3 inflammasome assembly at different stages. So, okay. I think we all agree that innate immunity is really important for our defense against pathogens, but it needs to be really regulated. So in case when there's a dysregulation, we get chronic inflammation. Also, for example, when there are endogenous triggers that interact with innate immune receptors. And this chronic inflammation can underlie many different pathologies. So I work, as I said, with inflammasomes. Inflammasomes are multi-protein complexes for activation of pro-inflammatory caspases. There are many different receptors that respond to intracellular threats. Upon detection of those threats, receptor oligomerizes. It then recruits adapter ASC. Adapter ASC makes this kind of specks close to the nuclei and acts for recruitment of inactive procase-bound molecules that get activated, and active caspase 1 is basically doing two different things. Like many more, but two different things that we are following. So one is that it can activate pro-inflammatory cytokines IL-1 beta and IL-18 into their mature forms. And on the other hand, it also cleaves protein called gasdermin. This cleavage releases N-terminal domain of gasdermin and can make pores in the plasma and other membranes. These pores can act as a conduit for the release of smaller proteins such as IL-1 beta and IL-18 and also HNGB1. But excessive poration leads to cell death called pyroptosis. And this is an example of pyroptotic dying cells, so we get this kind of ballooning-like morphology. So my focus is particularly on NLRP3 inflammasome, and this inflammasome is basically the most enigmatic. Why? I mean, there are many different things that can trigger it. So it can be pathogens, as we would expect, but there are also many endogenous activators, and for this session, especially amyloids are the ones that are important. There are also environmental factors that activate it, such as asbestos and silica in asbestosis and silicosis. But even prior, we knew what inflammasomes are. It was already known that mutations in the gene encoding NLRP3 were causing autoinflammatory syndromes called CAPS, cryopyrin-associated periodic syndromes. Cryopyrin is another name for NLRP3. So we also know that NLRP3 is regulated at many different levels. There are actually two signals needed for its activation. The first one, which we call priming, basically NF-kappa B transcription promotes production of NLRP3 protein and also pro-IL-1 beta. And there's the second signal that basically these activators trigger it, that induces assembly of NLRP3 protein and assembly of inflammasome, and this is basically the same cascade as I have shown in the previous slide. So, with all these different triggers from endogenous and exogenous origin, it's really no wonder that inflammasomes, in particular NLRP3 inflammasome, has been connected to many different diseases, including neurodegenerative diseases and cardiovascular diseases. So, if we look very briefly into how NLRP3 inflammasome might be involved in neurodegeneration. So a decade ago, we have shown that particular forms of prion proteins, such as fibrils or aggregates, were able to trigger NLRP3 inflammasome, but not the monomeric cellular form, neither oligomeric forms, which might be toxic, but they were not really inducing this pathway. But now, a decade later, basically, we know that there are many different pathological things going on in neurodegeneration that all might also contribute to NLRP3 activation. I will just as I said, protein aggregates, I will not be using in my study later really amyloids to trigger NLRP3, but I will be using silica and alum, which kind of activate NLRP3 in the same way as crystals and amyloids. So there is also increased ROS production, and ROS can basically act on NLRP3 at many different stages, and I will just kind of briefly touch that at the end. So the study that I will show today is basically we kind of got very naive idea. So most of you work with neurodegeneration, and you know that amyloids basically assemble from many monomers, let's say. And also what you probably know, that there were many attempts in the early thousands and so on to kind of inhibit amyloid formation by using peptides that kind of bind to monomeric forms or bind to protofibrils and kind of disrupt further assembly. So, I mean, we could do an analogy with inflammasomes. You have seen ASC-SPICs are huge. So these are also multimeric proteins. They are not amyloid in nature. They are mostly alpha helical and so on, but there are many different protein interactions that we could basically attack using our systems. So we got an idea, why don't we use different peptides that basically in sequence originate from components of NLRP3 inflammasome to kind of follow whether they could inhibit inflammasome formation or not. So, and these were the stages, so this was like in a bit, let's say 8 years ago, when no structure information was really present in the case of NLRP3. So we were really working in dark. I mean, we had no idea what we are doing in a way. So we have selected several peptides that originate in the sequence of NLRP3. Those were the ones that were kind of harboring important motifs for ATP binding in hydrolysis, like Walker A motif, Walker B motif, and sensor 1 region. But there were also, when we mapped CAPS mutations to the sequence, we saw that they are clustering in specific segments. So we also kind of made the peptides from those segments, just in case they could kind of be the ones that kind of promote oligomerization of NLRP3. So in this way, we wanted to inhibit oligomerization of NLRP3. Prior, it can basically recruit other components into the complex. So then we are also interested in blocking interactions between NLRP3 and adapter ASC. These are parent domain interactions. So we made two peptides, one that basically originates from NLRP3 and the other one that originates from ASC. Then we also wanted to block interaction between ASC and procaspase-1 molecules. So we used another peptide that basically originates from ASC but should inhibit this interaction. We used myristoylated peptides so that they were able to enter the cell, otherwise they would probably not be effective. So what we got from the peptides that originated from NLRP3, basically only two were having some inhibitory potential. This is, I'm not sure why it's really not seen that well, but this is Walker B, so you'll need to believe me. So it's Walker B and sensor 1 peptide, while others had no effect on silica-induced NLRP3. But we also used other triggers, so it's basically the inhibitory potential was the same, whatever trigger we used. So then when we go to the interaction domain peptides, here we actually had some information about the structures of those domains. So we were also much better in designing those inhibitory peptides. So basically all three that we had inhibited pretty well IL-1 beta release due to silica addition. So for now, we were only following IL-1 beta release, but we have also shown that basically these peptides already stop inflammasome formation at the step of ASC-SPIC formation, and of course then also caspases they inhibit the activation of caspase. So we were also interested, so far basically we were looking at canonical inflammasome activation, but we also were interested whether these peptides could inhibit CAPS associated constitutive activation. As I said, these mutations that cause CAPS basically make NLRP3 constitutively active, so you only need the priming step. You don't need additional trigger to get inflammation. We have we made a number of these models. So we use mouse macrophages that are deficient in NLRP3, and we introduce mutant or wild-type NLRP3 back to them. So, and when we induce the expression of NLRP3 by addition of doxycycline, we either do not get activation when we have wild-type, or we get constitutive activation in the case of CAPS mutants. We have 20 of those. I'm showing just 2. So basically, these peptides were able also to very well inhibit CAPS-associated constitutive activation. So then, of course, for us, it was also important whether these peptides are specific for NLRP3, or they are maybe just broad inhibitors of IL-1 beta release. So we were kind of hoping that the peptides that originate in NLRP3 sequence would be specific. They were not, which was a surprise. So we tested 2 other inflammasome. I'm showing NLRC4 inflammasome activation. NLRC4 responds to intracellular flagellin or type 2 secretion system proteins. And we also tested IM2 inflammasome that responds to double-stranded intracellular DNA. So basically, only one of the peptides that surprisingly originated from USC was specific for NLRP3 inflammasome. So then we worked a bit more with this specific peptide. So we made a scrambled peptide, which did not inhibit NLRP3 inflammasome activation. And then, so since this is a neuroscience conference, we were of course also interested whether we can get those peptides, especially this one that really kind of was specific for inhibition of NLRP3 into the brain. Peptides and proteins and other charged molecules are basically really notoriously known that they will not cross the blood-brain barrier. So we used a trick. So we tagged the peptide with 2 different domains. I'm showing one here. This is angio-PEP2 domain. So it's hydrophobic sequence that basically enables several proteins, such as aproteinin, to bind to the receptor on the surface of endothelial cells, and it can kind of induce transcytosis into the brain. So what you can't see, and what you should, is first that this peptide, even if it had the angio-PEP tag, was actually nicely inhibiting similarly to the non-tagged version. It could get very well into the cells, but so then we induced it IV into mice, and after 1 hour we sacrificed the mice and look at the organs where the fluorescence is present. So we were able to see that for the angio-PEP tagged peptide, there was a significant retention of the fluorescence signal in the brain, but not in the others. Other organs that we looked at, and for the non-tagged version, basically everything cleared pretty fast through the kidneys. So after 3 hours, there was still some retention for angio-PEP in the brain, but for the non-tagged version, there was basically no signal left in mice. So we also wanted to see whether these peptides could kind of work in vivo. So we use kind of a very simple model. So it's called silica-induced peritonitis. So we inject silica crystals into peritoneum, and then we follow how much neutrophil infiltration there is after several hours, and also how much IL-1 beta we get. And what we were able to show is that this peptide is basically able to inhibit silica-induced peritonitis in this mouse model. So I will just kind of briefly go through some of our recent studies, which are not really meant to kind of discover new inhibitors, but we did in a way. So we were interested. So I was now basically working on the really the early stages of NLRP3 inflammasome assembly. But as I have mentioned, if we look at caspase-1, we have this pro-inflammatory cytokine release, and we have pyroptosis, which might in some cases be even more problematic for the disease pathology. So we were interested, what really can we basically regulate Gasdermin D? Can we regulate pyroptosis? And we have found out in a genome-wide CRISPR-Cas screen that there is a regulator reg mTORC1 complex that basically through oxidative stress promotes Gasdermin D pore formation. So in this study, we have shown that if we inhibit mTOR activity, we can inhibit Gasdermin D pore formation and cell death, similarly to non-Gasdermin D inhibitor disulfiram. And on the other hand, what we could also do, we could just suppress oxidative stress levels, and also in this case, we get lower cell death. So I will just kind of briefly conclude with this busy slide, just more of an outlook. I mean, I'm not working that much on the inhibitor field. I mean, I'm interested in the mechanisms, but of course, NLRP3 has been kind of identified as a very important target for many different diseases. Particularly, there are some NLRP3 specific inhibitors that have entered clinic, like MCC950 derivatives, and also some others, like dapagliflozin, mostly for CAPS and also for cardiovascular disease. So I kind of, with all these different things that we could do, I'm kind of really hopeful that in the near future, if these trials kind of go well, that there will also be trials that will kind of tackle NLRP3 in some of the neurodegenerative diseases. So I would just like to conclude by thanking particularly to my PhD student, Dan Petrasusian, who did most of the work on the peptides, and also others who participated in that study. For the Gasdermin study, I'd like to thank Elvira Boršić and all these people, especially Jonathan Kagan and Charlie Evawolf. It was done, both studies were done in cooperation with them. I would like to thank my colleagues from the Department of Synthetic Biology and Immunology, particularly Professor Yarala, for his long-lasting support and my funding. So thank you for your attention.